Okay, we did our presentation on gene editing of embryos. Gene editing is a method to make changes to DNA by adding or removing DNA in the genome. It can be used on somatic cells or germline cells. It first started in 1996 with a sheep named Dolly, who scientists successfully cloned. More recently, CRISPR has been developed, which is a component of bacterial immune systems that can cut DNA. He Jiankyu is a Chinese scientist who illegally attempted to edit out HIV with CRISPR on two twin girls. They were the first and last gene-edited babies. CRISPR treatments are in trial, and in 2022, the Vertex Pharmaceuticals, which was a company based in Boston, brought their treatments to the regulators for approval, and that treatment was for sickle cell. There are many ethical concerns, such as the possibility of target effects and mosaicism and safety. Some people are worried that it is impossible to obtain informed consent because we are editing embryos. There is also a concern with gene editing only being accessible to the wealthy because treatments will be really expensive. Most stakeholders agree that it is important to have continuing public deliberation on whether or not germline editing should continue. As of 2014, there were 40 countries that discouraged or banned research on germline editing, including 15 nations in Western Europe because of ethical and safety concerns. There are risks and benefits to gene editing. Risks include concern regarding future generations. If we alter reproductive cells, modified genes could spread with unpredictable consequences. Some genetic traits could present harmful but could later prove pathogenic. It is impossible to tell the long-term effects of gene editing. However, some benefits that that gene editing could be used to prevent offspring from inheriting pathogenic genes. It could also be used for infertility treatments using non-obstructive sorry, azo- azoospermia in male patients and missense mutation in tube 8 in osteocytes in female patients. The discussion questions that we have are where do we draw the line between gene editing for treatment and gene editing for enrichment? Who would be liable for unintended results from gene editing? The patients, the researchers, or the government?
Okay, I'll be discussing health service needs and realities. So, genome editing of embryos come with many demands on the healthcare system. And so, first off, it includes healthcare professionals needing specialized training for gene editing technologies, such as for the CRISPR-Cas9. Especially with this being relatively new, it is crucial to stay up to date with these advancements to make sure that patients are treated safely. And so establishing regulations to ensure ethical standards are met and that there are safety protocols established to make sure patients are at less risk of harm. And then there are specific equipment and facilities needed to perform these procedures. So gene editing requires advanced labs and equipment to manipulate DNA in cells. And so in order to accommodate this, these requirements should be adapted. However, it could come at a pretty hefty price, which leads into the last demand, which are high costs. And so, although this may be an innovative and effective procedure, the costs would hinder it from being a normal option. And so, this is one of the main issues that needs to be addressed in order for there to be potential for it to become accessible in healthcare. And so, in the field of gene editing, several healthcare professionals play vital roles beyond the traditional responsibilities of doctors and nurses. And so, I'll be focusing on these three. First off is reproductive endocrinologists, who are key in managing fertility treatments and overseeing procedures like in IVF. And so they ensure that the body is prepared for gene editing, especially when creating embryos free from genetic disorders. Second, genetic counselors who help patients understand the genetic risks and benefits of gene editing, guiding them through complex ethical considerations and ensuring informed decision making. And so, lastly, there are embryologists who work in the lab directly handling embryos and performing the gene editing procedures. And so, they are responsible for ensuring that genetic modifications are made accurately, playing a crucial role in the success of these technologies. And so, the main concern is that leading CRISPR-Cas experts might be influenced by conflicts of interest, particularly if they have personal motivations tied to peer recognition or financial gain. And so these factors could unintentionally or intentionally shape how they present CRISPR-Cas based genome editing, its applications, and associated risks and benefits. And so while it's difficult to find direct evidence of bias, there should be a cautious approach toward trusting these experts given the challenges in fully understanding or ruling out potential conflicts. And so, for potential harm health providers could cause, given the surrounding off target effects, rushing to use germline editing for reproductive purposes could lead to unintended consequences. And so, including the introduction of genetic mutations or health issues in the child. And so, relating to beneficence, which requires acting in the best interest of the patient, health providers must carefully weigh the potential benefits of using genome editing to prevent genetic diseases against the risk of harm. And so, in the healthcare field, there's often pressure to adopt new technologies quickly, but this can create, as mentioned before, conflicts of interest when the focus shifts from patient safety to the potential profits or prestige of being the first breakthrough. And so Dr. Kubakova's study at the University of Oxford highlights why rushing into new methods like CRISPR-Cas9 can be dangerous. And so while CRISPR-Cas9 is efficient at targeting DNA, most cells repair the break in a way that causes mutations instead of fixing the issue. And so this shows how rushing to use such technologies without fully understanding them can lead to harmful outcomes, especially when the goal should be patient well-being over quick adoption. So, how providers can demonstrate sensitivity? Well, one is being transparent. And so, this includes discussing the potential risks and benefits of the procedure to make sure patients understand the long term consequences and excessive costs ahead. Another is being mindful of views and beliefs. And so, it is important to respect and understand that patients may have different views and values that may not really support the idea of genetic modification. And so, by having open conversations and communicating without judgment, it allows patients to feel more comforted about exploring other options. And so that kind of leads to a question to ponder about. As a healthcare provider, how would you navigate discussions with patients that ask to use gene editing for non medical enhancements?
Due to the restrictions placed on the gene editing of embryos, we don't have exact costs associated with this clinical scenario. However, we can look toward CRISPR-based gene cell therapy, generally priced at 2.2 million per patient for the US market. CRISPR treatments are expensive because pharmaceutical companies want to profit from their costly and potentially risky investments. Meeting the regulatory frameworks means that clinical trials cost pharmaceutical companies millions. The fact that these CRISPR cell therapies are intended to involve a single application may also lead pharmaceutical companies to raise their prices. The clinical success of several gene therapies has triggered the interest of how new investors and investment funds who are now turned their investments from other businesses to medical treatments because they seem economically profitable. These new investors may want to recover their investments with profits as soon as possible, thereby increasing the price of these gene therapies further. Additionally, CRISPR therapies are complex to manufacture and deliver. The manufacturing requires highly specialized staff, high-tech equipment, and practice-approved quality standards for safety and efficacy reasons. The delivery of CRISPR gene editing will require a complex healthcare infrastructure. Considering these factors, gene editing of embryos would likely have a multi-million-dollar price tag. These therapies would often be unaffordable for many national health systems, several private insurance plans, and the pockets for most potential recipients, making it incompatible with equitable access to the potential benefits of gene editing. On a global scale, such costly treatments reinforce inequalities between countries. Due to the high price of this technology, it is likely that this treatment would not reach the countries where disease is most prevalent and where many patients might be eligible for CRISPR treatment. While the market may be concentrated in high income countries, significant inequities can also exist within these nations, as reflected in the disparities in insurance coverage across different demographic groups in the United States. However, there are arguments that by curing or preventing a disease through gene editing of the embryos, the savings to health systems can be even greater than the price of gene editing process.
The realm of genome editing has multiple ethical concepts that can be put into consideration, and some of these include paternalism, which is defined as a patient having restricted autonomy. Therefore, they're deemed as incapable of making their own decisions and need a figure of authority to make decisions for them. In this case, an embryo cannot speak for itself and decide that it wants to undergo genome editing, so a parent or parents must make that decision for the embryo, and this could be the closest we could get to informed consent. A second term is beneficence, which means that the choices made should be in the best interest of the patient and be considered an inherently good decision. As to what is an inherently good decision is dependent upon the providers involved with the care. This is applied with genome editing, as the decisions made should ultimately be of benefit to the embryo, which again brings up the question of who decides what is beneficial and what is not, so that the embryo won't suffer any or have minimal risk of long-term consequences. The concept of justice is the idea that distribution for a service and a product may not be considered fair, as accessibility may be limited due to various factors, and genome editing can be considered a limited service, so therefore medical providers must decide who can receive or request the service and who cannot. And some of these factors that are included in making this decision can include service, reason for service, and price. The following terms deal more with the honesty provided to patients and the honesty in the practice that medical professionals provide. Veracity is the idea that medical providers should be honest when explaining the problems and solutions that come with a procedure or a service. When discussing the procedure of genome editing, providers should be honest and list the pros and cons so that parents are aware of what the consequences may be. And lastly, fidelity plays a part in making sure that medical providers hold accountability to the words and actions they offer a patient, in this case, the parents of the embryo. If a provider explicitly or even implicitly guarantees a certain outcome of the embryo to parents, then they should hold themselves accountable to that and ensure that they're able to meet that outcome.
HIPAA and GINA do not explicitly apply to the genome editing of embryos because they provide protection for genetic privacy of individuals after birth. Under the HIPAA 2013 amendment, genetic information was included in the definition of protected health information. Hypothetically, GINA would protect individuals who underwent germline editing from discrimination from health insurers and employers. Germline editing is not illegal in the US, but it is inhibited due to regulatory restrictions. In 2015, Congress passed an omnibus spending bill that explicitly prevented the FDA from using any of its resources to consider research applications involving germline DNA modification. Independently, National Institute of Health Director Francis Collins decided in 2015 that the NIH would not fund any use of gene editing technologies in human embryos, thus reflecting the intent of the Congressional Dickey-Wicker Amendment of 1996. The Dickey-Wicker Amendment prohibits federal funds for research in which a human embryo or embryos are destroyed, discarded, or knowingly subjected to risk of injury or death. Laboratory work using human embryos does not fall within the Institutional Review Board's jurisdiction unless the donors are identifiable, but this work may be overseen by voluntary oversight bodies such as embryonic stem cells research oversight committees or embryo research oversight committees. Considering the ambiguity in the restrictions against genome editing of embryos in the U.S., potential legislation reform would include stricter guidelines and penalties. For example, we propose the Embryonic Genome Act, which prohibits genome editing in human embryos within the United States. This establishes criminal and civil penalties for violations. Violations would include performing or attempting genome editing on human embryos that result in heritable modifications, as well as explicit prohibition of funding or sponsoring research with this intent. A 2020 review of the CRISPR global policy landscape found that the majority of countries studied do not have relevant legislation for the gene editing of embryos for non-reproductive purposes. Additionally, there are various degrees of restriction for heritable gene editing purposes. There are arguments for international legislation or consensus. These include mitigating medical tourism by traveling to less restrictive countries, technology governance to protect global human rights and medical and research ethics, and creating a shared vision of the future of gene editing technologies. Shared resources can reduce the need for every country to develop regulations. Most nations haven't developed regulations because it has not been a realistic undertaking until recent years. There are concerns for gene editing of embryos, such as disrespect to human heritage and challenging God's role. Additionally, there are arguments that gene-edited individuals cannot provide informed consent, which will extend to multiple future generations. Conversely, there is a potential perception of negligence if parents decide against performing gene editing. On a larger societal scale, there are concerns with the commodification of children, which can further exacerbate social inequalities, eugenics, and prejudice for individuals with disabilities. This disparity in access can be attributed to the high financial costs and varying regulations across countries. Additionally, there are potential unknown or unpredictable risks which could create novel modifications. A significant stigma of heritable human genome editing has noted that if one has the ability to modify the genes of their child, that is, to produce designer babies, there is a risk that the relationship between parents and children will undergo radical changes, with parents expressing their values, priorities, and aspirations in the genetic makeup of their children. Americans are closely divided over gene editing of embryos to reduce serious health risk. 49% of U.S. adults surveyed by the Pew Research Center reported that they would not want gene editing to greatly reduce their baby's risk of developing serious disease or conditions, while 48% reported that they would. However, 73% of U.S. adults surveyed stated that they would feel pressure to obtain this treatment for their baby. This exemplifies how societal pressure could exacerbate the desirability of gene editing of embryos. However, among religious U.S. adults, there is a stigma against this practice due to concerns of meddling with nature. Discussion questions include: you find out that your baby will have a genetic disorder that can be permanently treated through gene editing of the embryo. Would you want gene editing to reduce your baby's risk of developing the disorder? Would you feel pressured into agreeing to this treatment? Should there be a complete ban on gene editing of embryos? Should there be different restrictions based on whether or not it is for reproductive purposes?
So, no matter what, a nurse should have confidentiality and privacy practices involved in the care they provide. But along with this, they should also have competent knowledge in the field of genome editing so that they may be able to answer questions and address concerns parents of the embryo may have. This can be done by having the proper qualifications and staying up to date with new technological advances and studies that could alter the current procedure of genome editing. Delivering news empathetically is already a requirement when dealing with patients, but more so in this field because it could result in negative consequences that should be delivered in a direct yet empathetic manner, especially considering that this field is seen as controversial or can be deemed as controversial. An important idea is that, in general, any medical procedure, service, drug administration, and etc., is done so with informed consent. So it is a nurse's obligation to inform the parents, the patients, in this case, the parents of the embryo, of what they're agreeing to do. They should be aware of the risks genome editing poses, which can be confirmed by asking questions. So these ideas of competency, empathy, and ensuring there is informed consent would be applied to nursing practices, and it should be done so regardless.
The field of genome editing can be seen, as mentioned before, controversial. Therefore, I would advise nurses that have ethical views and opinions that don't align with the procedures, ethics, or ideas of genome editing to not consider this a field in which they'd want to work in.
I did the case study, and I think it's important to look at case studies, specifically in gene editing, as these cases can tell us how to look at these different situations in the future and how we can go about dealing with them. So the case study is a couple, AB male and ED female, want to have a baby. AB, the male, is an HIV CCR5 gene carrier, and ED is not. The couple does not want their babies to be born with HIV, as many don't want that, right? A researcher conducting unsupervised research claimed that he could render the babies immune to HIV infection through CRISPR-Cas9 technology, making them the world's first gene-edited babies. After ED, the female gave birth to the twins. The researchers didn't provide full details about how they collected and analyzed their DNA samples. This experimental procedure was shown off in the conference and was deemed irresponsible by the International Science Committee due to its lack of peer review, proper regulation, and data transparency. It is unknown whether there were off-target effects, consequences of CCR5 gene manipulation, or any potential benefits for the twins. The researcher was found guilty of conducting illegal medical practices and sentenced to 3 years in prison. The stakeholders in this case were the twin girls. They are subject to the potential health risks of the gene editing of the embryos and the potential of no benefit of HIV immunity. Secondly, the parents of the twins have potential psychological consequences due to controversy, potential lack of informed consent for this experimental and unsupervised research. Then thirdly, the scientists and researchers, they have the responsibility of upholding international ethical consensus, research procedures, and discussions regarding the future of gene editing of embryos. Lastly, the government legislation can place greater restrictions and guidelines for gene editing research with embryos. Some questions to think about is, given that safe and effective methods already exist to prevent HIV transmission from parent to child, was there any justification for editing the CCR5 gene in the researcher's experiment? How should the availability of alternative treatments factor into ethical decisions about gene editing? How do the ethical principles of beneficence, non-maleficence, and autonomy play into this case study? Thirdly, if the twins grow up healthy and immune to HIV, does this validate the experiment, or do ethical concerns and risks outweigh the results? What does the backlash of this case reveal about international sentiment and the future of gene editing of embryos? Lastly, we have our resources here, and then that's the end. I hope you enjoyed our presentation, and thank you.